Dear honored colleagues, thank you for listening to my presentation. It is a great honor for me to be invited to present in 2023 AMMF's Cholangiocarcinoma Conference. My name is Kefei Yuan. I'm from West China Hospital, Sichuan University, China. Today, I would like to present you with updated advances of circular RNAs in cholangiocarcinoma. Circular RNA is a type of single-stranded RNA that is rapidly emerging in RNA research field. Unlike traditional linear RNAs, which has 5 terminal and 3 terminal ends, circular RNAs are covalently closed single-stranded RNAs. This feature confers numerous properties to circular RNA, many of which have only recently been identified. For example, it is resistant to RNA exonuclease-mediated degradation and is presumably more stable than most linear RNA in cells. The fact that single-strand RNA could be in a circular form was first identified in plant spheroids in 1976, as evidenced by this electron micrograph. After that, this kind of circular transcript was also discovered in eukaryotic cells in 1979. However, at that time, the function of circular RNAs were completely unknown. Therefore, for a long time, they were regarded as byproducts of post-transcriptional mRNA modification or splicing errors. Over the last 10 years, the advance of RNA sequencing technologies combined with computational pipelines for circular RNA annotation have revealed widespread expression of circular RNAs in a variety of metazone cell types and tissues. Now we know that the majority of annotated circular RNAs are produced from pre-mRNA backsplicing. Pre-mRNA backsplicing utilizes the spliceosomal machinery and is regulated by intronic complementary sequences in flanking introns of circle-forming exons and RNA binding proteins that can bind to flanking intronic sequences. The RBPs can enhance or impair intronic ICS pairs, leading to increased or dampened circular RNA expression. RBPs can also directly bridge distal splice sites to enhance the backsplicing. With the identification of more and more circular RNAs, the functions of circular RNAs are gradually elucidated. It has been demonstrated that circular RNAs can serve as RNA aptamer, sponges of microRNAs or proteins, therapeutic targets or biomarkers, regulators of innate immune response, and last but not least, although circular RNAs are widely known as a type of non-coding RNAs, some of them have been proved to be able to code proteins. Circular RNAs have been shown to regulate the progression and metastasis of many different types of cancers, including cholangiocarcinoma. A series of circular RNAs are reported to either suppress or promote the progression of intrahepatic cholangiocarcinoma and distal cholangiocarcinoma. However, until now, although perihilar cholangiocarcinoma accounts for 50 to 60% of all CCAs and is the most common CCA, no publication has reviewed the involvement of circular RNAs in PCCA. Next, I will present you with a few typical examples of circular RNAs in CCA according to their distinct mechanism. The competing endogenous RNA hypothesis was first proposed a decade ago, which states that RNAs can manipulate biological functions independently of protein translation. Many circular RNAs have been revealed to function through the CERA network, which means that they can either serve as microRNA sponges or protein sponges. As microRNA sponge, circular RNA releases mRNA from the binding of microRNAs and promote the translation of mRNA. On the other hand, as RNA binding protein sponge, circular RNA strengthens the interaction between microRNA and mRNA, which blocks the translation of mRNA. Also, in this manner, circRNA can regulate the interaction between proteins. Here is an example of circRNA functions as sponge for microRNA. circACTN4 acted as sponge of miR4245P, which downregulates the level of free miR445P. YAP1 mRNA is the target of this microRNA. Therefore, decreased free miR4245P levels led to upregulation of YAP1 expression and activation of hippo-YAP signaling pathway, finally contributing to iCCA growth and metastasis. Next, I will introduce an example of circRNA serves as sponge for proteins. This is a paper of our lab. In order to screen for essential circRNAs contributing to iCCA metastasis, circRNA sequencing was performed to analyze the differentially expressed circRNAs between primary iCCA tissues from patients that experienced extrahepatic metastasis after surgery and primary iCCA tissues from patients who do not experience postoperative metastasis or recurrence. As the most abundant differentially expressed circRNA, circNA5B was chosen for further study. After that, we showed that circNA5B could inhibit the proliferation and metastasis of iCCA cells, both in vitro and in vivo. To dissect the molecular mechanisms of circRNAs in suppressing iCCA cell proliferation and metastasis, we performed RNA sequencing to identify the potential genes regulated by circNA5B knockdown. And KEGG pathway analysis showed that MAPK signaling pathway was one of the most remarkable pathways enriched. As aberrant activation of MAPK signaling pathway is reported to be involved in oncogenesis and metastasis of various tumors, we postulated that circNA5B inhibited iCCA growth and metastasis via regulation of MAPK signaling pathway, as confirmed by our immunoblot results. Overexpression of circNA5B repressed ERK phosphorylation, while the phosphorylation levels of P38 and JNK were not altered. These two are the other two K kinases of MAPK signaling pathway. Our data suggested that circNA5B might inhibit iCCA growth and metastasis by regulating the ERK phosphorylation. To elucidate how circNA5B regulates the phosphorylation of ERK, a bionated circNA5B probe was used to perform RNA pull-down in iCCA cell lysis, followed by mass spectrometry analysis. Strikingly, the mass spectrometry results show that the MEK1, the kinase that phosphorylates ERK, was likely to interact with circNA5B. Then, through a series of rescue experiments, we proved that circNA5B competitively bound to MEK1 and prevented the interaction between MEK1 and ERK by sponging MEK1, thereby inactivating ERK phosphorylation, leading to downregulation of the target genes of ERK, such as CCND1, MPNP1, etc., and decreased expressions of this gene that promote cell proliferation and metastasis finally contributed to repression of tumor growth and metastasis of iCCA. After introduction of circRNAs as sponges for microRNAs and proteins, now I will present you with another paper of our team, which identified a protein-coding circRNA, the circGGMBP2. circGGMBP2 is a IL-6-induced circRNA that can promote the tumor growth and metastasis of iCCA. To examine the protein coding potential of circGGMBP2, we first analyze the sequence of this circRNA to find out whether it has a putative open reading frame and IRES elements, which are required for protein coding. Interestingly, we identified the IRES elements and a highly conserved ORF with a potential of coding a protein of 184 amino acids. To verify whether circGGMBP2 encodes a protein of 184 amino acids, we performed SDS page with cell lysis of circGGMBP2 overexpression iCCA cells. As predicted, an enhanced band could be observed after circGGMBP2 overexpression compared with the control vector on a SDS page gel. Subsequent mass spectrometry confirmed that the amino acid sequences were highly aligned to the putative sequences of CGGMBP2-184AA, the potential novel protein that was coded by the circRNA. To further characterize CGGMBP2-184AA, we generated a highly specific antibody against the amino acid sequences of this novel protein, which was supposed to recognize both GGMBP2, the parent gene of circGGMBP2, and CGGMBP2-184AA, the novel protein. Importantly, the expression of CGGMBP2-184AA was drastically upregulated after circRNA overexpression, whereas GGMBP2 expression level showed no significant change. These results demonstrated that circGGMBP2 indeed encodes a protein CGGMBP2-184AA. After that, we performed a series of experiments to prove that CGGMBP2-184AA interacts with STAT3, phosphorylates its tyrosine 705 site, and initiates the transcription of the downstream target genes of STAT3. And the IL-6 CGGMBP2-184AA STAT3 formed a positive feedback loop to sustain iCCA growth and metastasis. We also confirmed that circGGMBP2 regulated cell proliferation and metastasis of iCCA mainly through the protein CGGMBP2-184AA rather than its circular RNA form. And finally, I want to talk about the clinical applications of circRNAs in CCA. A series of studies have demonstrated the prognostic value of circRNAs. For instance, our study found that circNA5B was significantly downregulated in primary human iCCA tissues compared with normal tissues, and patients with lower circNA5B expression had worse overall survival and recurrence-free survival. Likewise, we also showed that higher circGGMBP2 expression was associated with shorter overall survival and recurrence-free survival after curative resection in iCCA patients. In addition, circRNAs are often expressed in a more cell-type and tissue-specific manner. Thus, circRNAs may have advantages over microRNAs and long non-coding RNAs as biomarkers for diagnosis of CCA. However, the diagnostic value of circRNAs in CCA remains to be validated. CCA patients are usually diagnosed at an advanced stage, and almost all of these patients require chemotherapy. Active chemotherapy regimens of CCA include cisplatin, gemcitabine, and capecitabine. Here, SMARCA5 has been shown to increase the sensitivity of CCA cells to cisplatin. And also, it has been shown that this circRNA can increase the sensitivity of CCA cells to gemcitabine also. Moreover, high levels of circRNA has been shown to delay cancer cell resistance to trametinib, a MEK inhibitor which has the potential to be used as an anti-CCA drug, which has been evidenced by recent studies. To sum up, although the aberrant expression of circRNAs has been shown to be associated with the progression and chemotherapy resistance of CCA. The roles and mechanisms of CCAs in the development of CCA remain unknown. Additionally, some preliminary drug sensitivity tests have suggested that CCAs may be promising new therapeutic targets of CCA, but further studies are needed to confirm their reliability and clinical reproducibility. And we believe that further study of CCAs will provide more evidence and novel insights into the pathogenesis of CCA. Understanding the roles and molecular mechanisms of CCAs in CCA is of great clinical significance for improving the survival rate and reducing the postoperative recurrence rate of CCA patients. That is all for my presentation. Thank you very much for your attention.